that will be on Cadazil and Carazil and will be presented by Dr. Haffner from Munich. Dr. Haffner?
Hope I'm not going to drop this at some point. Okay, thank you for the introduction. I would also like to thank Marie-Germain Bouzet and the organizers of this very beautiful workshop to invite me and to have a presentation at this very special setting. Since we have already heard a lot about Carazil today, I decided to focus my presentation on Carazil. To find out this, okay, this is the one. And what I would like to do is I would give you a brief overview of the history of the first Carazil cases and a brief description of the pathology. And then I will focus the main part of my talk on molecular mechanisms that underlie this disease. And when Marie-Germain gave her introduction at the very beginning of this day, I realized that the first Carazil patient might have been much farther back in history than the first Carazil patient, because what is assumed to be the first Carazil patient is a 30-year-old man that was seen at the neuropsychiatry department at the Tohoku University in Japan. And since then, Carazil has mainly been, or until very, very recently, Carazil has been more or less a Japanese peculiarity because the major, or the main, the large majority of patients have been seen in Japan. And this patient died soon after an autopsy was performed, and the results from this autopsy and from the autopsy of two unrelated cases were described in 1976. And this is the first comprehensive pathology description of this disorder, and it was at that time called familial encephalopathy of Binswanger's type without hypertension. Now, in the 1980s, several other reports about patients were described and described with showing progressive leukoencephalopathy, but also alopecia and spondylosis, which have now in the meantime been or are now considered very important diagnostic features. In 1987, the recessive inheritance pattern of this disorder was recognized. And in the 1990s, the first MRI images of Carazil patients were shown or were published, and they showed widespread, diffuse, and extensive white matter hyperintensities. Now, oops, sorry. In 1994, the term Carazil was proposed, and in contrast to the name finding for Cadazil, there was not a lot of discussion because it was just assumed that it is a related recessively inherited form of Carazil, so the termination was very easy here. And it also referred to the similarities to the pathology to Carazil, including the white matter hyperintensities, of course, and the lack of hypertension, but also to the similarities in histopathology, and this refers to the loss of smooth muscle cells in both of these diseases in cerebral brain vessels. Our major breakthrough then occurred in the year 2009 when HTRA1 was identified as disease-causing gene. And this was done on the basis of linkage analysis in 5 patients, so not 50 patients, but 5 patients. And so this linkage analysis identified a candidate interval of 2.44 megabases on the chromosome 10. And within this genetic interval, HTRA1 appeared to be the most promising candidate mainly because of its expression pattern in vessels, bone, and skin, which reflected best the symptoms, including alopecia and spondylosis. HTRA1 is a serine protease that is highly conserved in eukaryotes and even prokaryotes. And earlier publications had implicated this protease in the transforming growth factor beta signaling pathway. So mainly these two publications proposed that HTR1 is an inhibitor of TGF-beta signaling. So it appeared a likely candidate because for a disease-causing gene for Carazil, because TGF-beta has long been known as major player within the microvasculature. Deregulation of the TGF-beta pathway has been shown to cause diseases like Marfan syndrome, the related Loeys-Dietz syndrome, fibrotic disease, pulmonary arterial hypertension, and hereditary hemorrhagic telangiectasia, which is characterized, among others, by arteriovenous malformations. So it seemed plausible that TGF-beta might also be involved in Carazil pathology. Now, of course, that had to be proven, and indeed, in all of these 5 patients, mutations in the HTRA1 gene were identified, and the majority of them located within the catalytic domain. And the activity of these mutations were analyzed in an in vitro assay using a fluorophore in vitro substrate, and it turned out that all but one of these mutations showed strongly reduced activity compared to the wild-type protein, and this activity was almost as low as an active site mutant, which is expected to completely abolish proteolytic activity. Then there was one mutation, this one, a nonsense mutation that located slightly outside this domain, and this showed in this assay full activity, but it was later shown to cause an mRNA decay in these patients. So in total, the analysis of this, or the proteolytic analysis of all these mutants strongly indicated that we really have a loss of function mechanism here. Then, of course, the effect on TGF-beta signaling was analyzed in a cellular luciferase assay, where a luciferase plasmid, or where luciferase plasmid was used under the control of a TGF-beta dependent promoter, and it could be shown that when in these cells TGF-beta signaling was induced by external addition of TGF-beta, there is a strong increase in signaling, which can be blocked by the wild-type protease. But none of the 3 tested mutants were able to do so, strongly indicating that they are really loss of function, and they can also attenuate, or let's say that they have lost their ability to attenuate TGF-beta signaling. This was in line with data from Carazil patients. For example, an increased expression of TGF-beta in brain vessels, and with data from fibroblasts from one of these patients, which showed an increase in expression of noggin mRNA, which is an established TGF-beta target gene. So altogether, these data suggested that TGF-beta signaling is indeed contributing to Carazil pathology, and that it's probably, or it appeared that Carazil signaling is increased in these patients. TGF-beta is normally produced as a pro-protein that is intracellularly processed and secreted as an inactive complex together with latent TGF-beta binding protein 1, a structural protein that mediates the sequestration of this whole complex within the extracellular matrix. And there, the protein is attached until the signaling is triggered by a variety of signals. According to the data I've just shown you and of additional data that had been done in cellular systems, the Japanese authors proposed the following mechanisms how HTRA1 could attenuate the TGF-beta signaling pathway. So they proposed that HTRA1 already within the cell cleaves the prodomain of TGF-beta, and this leads to a degradation of the complete TGF-beta molecule by the endoplasmic reticulum associated degradation mechanism. And this, of course, would lead to a reduced level of TGF-beta available within the extracellular matrix. Now, this mechanism primarily relied on in vitro data from overexpressing systems. And in addition, it disregarded the fact that HTRA1 is a mainly secreted protease. So this model was a little bit was in contrast to other or these other observations. And at that point, we got interested in analyzing this model a little bit further. And this was a project that Nathalie Beaufort in our lab, a postdoc in our lab, started. And she first developed an HTRA1 proteolysis assay to verify if HTRA1 is really degrading TGF-beta. And what she did, she took purified HTRA1 and combined it with supernatant from cells overexpressing TGF-beta1. And indeed, she could find a reduction of mature TGF-beta, as seen here on this immunoplot, however, only at rather high protease concentrations. And when she used in this assay another substrate, namely the LTBP1 protein, she could also find a processing, but at protein concentrations as low as 1 nanomolar. So this indicated that LTBP1 might be a much more efficient target than TGF-beta itself. We moved on and asked the question, do Carasil mutations affect the processing of LTBP1? And for this, they modified the assay by combining two supernatants from two differently transfected cells, one medium containing LTBP1, the other medium containing either wild-type HTRA1 protease or one of these three mutants, Carasil mutants or the active site mutation. And what she could find is that with the wild-type protease containing supernatant, LTBP was indeed proteolyzed, whereas none of the other mutants were able to do so, although they were secreted efficiently into the extracellular medium. So it was not a problem of the secretion of the protein, but of their activity. Next, we wanted to map the cleavage site within the LTBP1 molecule a little bit closer. And for this, we constructed an expression LTBP1 molecule with tags at both ends. And processing of this molecule with HTRA1 and detection of the C-terminal fragment led to the appearance of a very distinct band with a lower migration behavior, whereas the N-terminal fragments appeared as an array of bands. This indicated that the cleavage occurs within the N-terminal region of the LTBP1 molecule, and we could indeed verify this and even map the cleavage site more precisely by using now a truncated version, a C-terminally truncated version of the LTBP1 that was expressed and proteolyzed by HTRA1. And Nathalie then isolated this cleavage fragment and subjected it to amino acid sequencing. And the sequences we yielded, or which we obtained from this experiment, corresponded to a region indeed within the N-terminal part of the LTBP1 molecule, and they moreover were part of a region that had previously been identified as fibronectin binding domain of the LTBP1 molecule. So this indicated that the cleavage might have some functional consequences. And of course, we wanted to test this. And Natalie then established a solid phase binding assay to test this association between these two molecules by immobilizing fibronectin in a microtiter plate and incubating a supernatant, an LTBP1 containing supernatant, and quantifying it by a colorimetric assay. And indeed, what she could find, whereas the untreated LTBP1 molecule showed a nice binding, a strong reduction in binding was observed upon treatment with wild-type protease, whereas treatment with the active site mutant protease did not show reduction in the binding. And here the appearance of the cleavage fragment is nicely seen. So there was, at least in this assay, a clear functional reduction of the interaction. And this could be reproduced in an assay where we used an LTBP1 construct that exactly corresponded to the cleavage fragment. So this is not a cleaved LTBP1, this is a fragment that corresponds exactly to the cleavage product, as seen here. The running behavior corresponds to the cleaved protein, and also here the binding the interaction capacity of this construct versus the wild-type protein is strongly reduced. Now, still, this was a rather artificial assay. We wanted to perform it in a more physiological setting. What we did is to replace the immobilized fibronectin by an extracellular matrix produced by primary aortic smooth muscle cells. So we cultured these cells, let them produce the extracellular matrix, and the binding assay performed was similar to I have shown before. This matrix produces high amounts of fibronectin, as seen on this immunofluorescence staining. And when Natalie performed the binding assay, she could see a very similar result as shown before. LTBP that had been treated and processed by HTRE1 protease showed a reduced binding to this extracellular matrix. Now, in our next step, or maybe I can go back, this now clearly showed that there might be functional consequences of HTR1-mediated cleavage of LTBP1. However, up to this point, we also had used overexpression systems. So we wanted to find out if also cleavage occurs under endogenous HTR1 expression conditions. And for that, Eva Scharrer, a PhD student in the lab, made use of the HTRE1 knockout mice that had before been generated by a gene trapping approach. So sequencing of the genetic locus identified the insertion site of this trapping construct within the intron 1 of the mouse HTRE1 gene. And this insertion of this trapping vector led to a misplicing of the primate transcript, and these mice express a splice or a mature transcript that lacks all exons but the first exons of the HTRE1 gene. Analysis of the HTRE1 mRNA in brain material demonstrated the absence of this transcript. And by the way, these mice are viable, they are fertile. So the homozygous knockout mice, they are viable, fertile, have a normal life expectancy, and they show no obvious phenotype. We are currently analyzing whether they have a more subtle phenotype, but at the moment we cannot give you a conclusive answer about the phenotype of these mice. However, what Eva did, she isolated embryonic fibroblasts of these mice and immortalized them by serial passaging. And this graph shows the proliferation behavior of these two lines, of a knockout line and a wild-type line. And using supernatants from these immobilized fibroblast lines, we were, for the first time, able, as seen here, to demonstrate the absence of HTRA1 protein in this model by Western blotting. So here, two wild-type lines show a nice signal, which are absent in the knockout lines. When these supernatants with endogenous HTRA1 were now incubated with LTBP1, they produced a similar cleavage fragment as shown before, demonstrating that cleavage can also occur under endogenous expression conditions. We then used a HTRA1 specific inhibitor to find out if LTBP might be cleaved by other proteases in the system, and apparently it's not, because addition of this inhibitor to this assay completely inhibited the processing of LTBP, demonstrating that at least in this cell type, HTRA1 is the primary LTBP producing protease, which was an important finding. Then we had a system now in our hands to analyze if HTRA1 deficiency is really affecting TGF-beta signaling. And in the first approach, we used supernatants from these fibroblast lines and measured by an ELISA assay the TGF-beta protein levels. And by using 7 to 8 of these lines and combining them in a statistic, we saw a significant decrease of TGF-beta levels in the knockout cell lines. Then we wanted to know, does this reflect a difference in bioactive TGF-beta? And for this, we established a bioassay that involves a cell line that has been stably transfected with a reporter plasmid that encodes a soluble alkaline phosphatase, an enzyme that is under the control of a TGF-beta dependent promoter. So this cell, to this cell line, TGF-beta is added, the production and secretion of this alkaline phosphatase, which is now TGF-beta dependent, can be measured by a chemiluminescent assay. And when we used these cell lines and treated them with supernatants from our immortalized fibroblast lines, we could nicely show a strong reduction, a significant reduction of bioactive TGF-beta in the supernatants of the knockout lines of the HTR deficient cell lines. So this fitted nicely to the ELISA data that I've shown before. Then, of course, we wanted to go one step further and to look at the signaling itself. And TGF-beta signaling, you see the main components of the signaling pathway are depicted here. Mature TGF-beta binds to a heterodimeric receptor. The signal is relayed by phosphorylation to intracellular transducers of the SMAD family, and eventually these complexes move to the nucleus and alter transcription of target genes. Now, one of the established measurements, or of the established and most established monitoring assays in this pathway is the detection of phosphorylated SMAD proteins, and you see this here nicely in an experiment done in our wild-type cell lines. When a recombinant TGF-beta is added, you see a strong increase in the phosphorylation of these proteins. Now again, this was done with recombinantly added TGF-beta. We now repeated this experiment with supernatants from our MEF cell lines. And indeed, what we could see, supernatants from wild-type cell lines showed a significant increase in phosphorylation, or it showed a phosphorylation, whereas the signal was much weaker, or strongly reduced with supernatants from the deficient cell lines. And this is a control immunoplot showing that the protein amount in these cells was equal. And when we combined all the data in a statistic, we could see that there is a significant reduction in this phosphorylation of this MAD protein within the knockout cell lines, further supporting or further suggesting that TGF-beta is indeed affected in these lines by HTR1 deficiency. Now then, finally, we wanted to analyze TGF-beta target genes. And for that, we chose to establish target genes, the PI1 and the CTGF. And this was now done within the cells. So we analyzed we quantified mRNA levels of these target genes by real-time PCR. And what we could see a strong reduction. So this was again done in our 7 to 8 cell lines of each type, of each genotype. And we could see a strong reduction in PI1 expression and also of the CTGF-beta reduction expression in the knockout cell lines, strongly suggesting that there is a loss of TGF-beta signaling upon HTR1 deficiency. So these data fitted very nicely to the data I've shown before from the ELISA and from the active TGF-beta tests. And we would now like to propose that HTR1 is indeed affecting the TGF-beta signaling, and that the cleavage of LTBP1 might be a crucial step in the pathology of CARASIL. So let me summarize the data I've presented you. As said, from the literature, it was known that CARASIL patients carry homozygous HTR1 loss-of-function mutations and show signs of enhanced TGF-beta activity. Now, we now would like to suggest that the latent TGF-beta binding protein is a major HTR1 substrate, and that it could be the major mediator of the deregulation of TGF-beta signaling within CARASIL. And we basically confirm data that have been hypothesized before, that the deregulation of the TGF-beta pathway might represent a key pathological mechanism in this disorder. And this is now also, let's say, complemented by data from the work of Anjutel, who has identified LTBP4 as well as HTR1s as components in NOTCH3 aggregates. So this now would provide a link between these two diseases and suggests that there might be a common disease mechanism for both disorders, and that could possibly involve the TGF-beta signaling pathway. And then, of course, in the long term, the final question would be, or one of the major questions would also be, if this is a mechanism that could also be relevant for sporadic small vessel diseases. And with that, I would like to thank the people that were involved in this, mainly Nathalie Beaufort, who is here in the audience, and Eva Schara, other members of the lab, and our collaborators, Anjutel, with whom we started a very fruitful collaboration, the members of the Leduc network, and I would like to thank you for your attention.